So good day everyone. So today we are tackled about nucleotide, blast or BLASTn. So first, what is BLAST? In bioinformatics, BLAST or Basic Local Alignment Search Tool is an algorithm for comparing primary biological sequence information, such as the amino acid sequences of proteins or the nucleotides of DNA and or RNA sequences. A BLAST search enables a researcher to compare a subject protein or nucleotide sequence, called query, with a library or database of sequences and identify library sequences that resemble the query sequence above a certain threshold. BLAST can be used to infer functional and evolutionary relationships between sequences as well as identify members of gene families. Next is the background. Before BLAST, FASTA was developed by David J. Lippmann and William R. Pearson in 1985. BLAST and FASTA are two similarity searching programs that identify homologous DNA sequences and protein based on the exist sequences similarities. They provide facilities for comparing DNA and protein sequences with the existing DNA protein databases. FASTA is pronounced FASTA and stands for FASTall because it works with any alphabet, an extension of the original FAST-P or protein and FASTn for nucleotide alignment tools. BLAST came from 1990 stochastic model of Samuel Karlin and Stephen Altshull. Subsequently, Altshull along with Warren Gish, Webb Miller, Eugene Mears, and David J. Lippmann at the National Institute of Health designed the BLAST algorithm, which was published in the Journal of Molecular Biology in 1990 and cited over 75,000 times. Next is the uses of BLAST. First is identifying species. Second, locating domains, establishing phylogeny, DNA mapping, and last is comparison. Next is program. Nucleotide BLAST or BLASTn. This program, given a DNA query, returns the most similar DNA sequences from the DNA database and the user specify. Next is protein BLAST or BLAST-P. This program, given a protein query, returns the most similar protein sequences from the protein database that the user specifies. Next is the BLAST homepage and selected search pages. So I will introduce the BLAST homepage and form elements or functions of selected search pages. So as you can see, there are a lot of arrow letter in the picture of BLAST homepage. So the background of BLAST is a suite programs provided by NCBI for aligning query sequences against those present in a selected target database. The NCBI BLAST homepage provides an access point for these tools to perform sequence alignment on the web. So the BLAST homepage. So there are a lot of several sections. So each provides a specific set of functions. So first arrow letter A is the common header. It present in most BLAST related pages, provides easy access to other content or functions not directly accessible from the homepage. Second is the recent result. So it provides links to a list of recently completed results, BLAST related news and search tips. So next is there are three links are organized into three categories, with pages with web forms for submitting searches are listed as links in the body of the BLAST homepage. So first is the arrow letter C is the web BLAST. Second is the BLAST genomes, and third is the specialized BLAST. Next is the search box or the arrow F. So in the BLAST assembled genomes section takes the name of an organism as input and suggests a list of candidates. Selecting from the suggested list will locate the best genomic sequence data set for BLAST alignment purposes. So next picture is the common BLAST header. The common BLAST header provides a convenient way to navigate among different pages to access different contents or functions. First is the NCBI logo. So it links to the NCBI homepage to allow access to non-BLAST related functions and content. Next is the home tab or the letter H. So it direct links to the BLAST homepage. So BLAST search results are temporarily saved for 36 hours. So in recent results or the letter I, it links to a page that keeps track of recently submitted search requests that have not expired. The request ID uniquely assigned to a submitted search provides a one-click access to the result. Next strategies tab. So list a set of search set up saved earlier. It allows the examination of search settings, use quick relaunch of these searches, and download of specific strategies for sharing out or reuse in standalone BLAST. Next is the Help tab. Points to page with a list of links to help documents, tutorials, references, and useful download directories on the BLAST site. And the last is My NCBI. So it is a free account from NCBI. So which allows user to customize their site preference and manage their works performed on the NCBI site. Login related links for NCBI or the letter L are at the right. BLAST searches performed by login to My NCBI accounts enable access to BLAST search results for their full 36-hour lifespan through the Recent Results tab. Strategies saved will be saved permanently. So next is the Recent Result page. So BLAST search results are available for 36 hours. So the Recent Results tab display a list of recently submitted search requests that have not expired. The list is session specific and will be lost if session cookies is cleared upon browser exit. For this reason, it is recommended that BLAST searches be done with an active login to a My NCBI account. So first is the My NCBI. The My NCBI login from the header is shown as an insert at the upper right. So each result is given as a row in the table. Next is the identifier in the request ID or RID column. So it provides an one-click access to the search result. Next is the program, title and database column or the letter C. So it combines to provide a summary for a specific search. So next is the letter D, the pop-up upon mouse over. So that is the database restriction applied are indicated by more. Next is the save, download, and the read X. So save, it allows saving the search strategy. Second, downloading the search strategy. And the third, the read X, is removing the search from the list. Next is the letter F. So the input box above the table is for retrieving other results using their assigned RIDs. So such as those shared among colleges used as teaching or demonstration examples, or those with issues encountered and reported to NCBI's BLAST Help group. So by clicking the Go button without an RID loads, so the top is the letter G, that is the format form, and letter H is where the content, and I is the format of result can be adjusted. Next is the Save Strategies page. So the Save Strategies tab displays a list of search strategies. So the first 4 columns, the letter G, provides a good summary of the search settings for each save entry. Second is the view link, or the letter K, loads the settings in a search page, while the download link, or the letter L, saves the settings in an ASN. So in choose file and view button, letter M, one formatted file for use with standalone BLAST or reloading on the web services using the choose file. So next is the letter N. By clicking the red X, so it removes the entry from the list. Next is the elements of the standard nucleotide BLAST search page. So the nucleotide BLAST link loads the standard nucleotide BLAST search page. So first is the letter A, or the top of the page. So it contains the breadcrumb indicating the page position in the site hierarchy. Next is the letter B, the page title, a set of tabs for quick navigation among the 5 core BLAST search pages. So next is the letter C, the receipt and the bookmark. So in letter C, it is links to set the page back to default and bookmark a search page with customized settings. So the default display of the page contains the 3 sections with the functions described below. So in enter query sequence, so the letter D, the main input box, takes nucleotide query sequences in various formats. So for a single query, or in the letter E, the query subrange boxes, it defines a segment of the query to be used in the search. Query sequences saved in the plain text file can be uploaded using the choose file button, or the letter F. So next is the align 2 or more sequences check boxes, the letter J. So changes the choose search set section below to enter subject sequence to allow comparison between the query and those in the subject input box. Next is the choose search set. So first is the letter I. So BLAST database can be selected from the standard list using the pull down menu. So second is the letter J. So a search can be restricted to a subset of entries in the selected database using the organism field by typing a name of the species, strains, or taxonomic group in the text box and selecting from the suggested list. Next is the letter K. Exclude optional, the models XM or XP. So checking the exclusion box to the right exclude sequences from the selected organism from the search. Multiple organisms can be selected by adding extra input box using the plus button. Specific types of relatively low value sequences can be excluded using the check box below. So next is the letter L, or the last sequence from type material. So for certain databases, entering custom queries in the entries query text box, it will restrict a search to entries satisfying the specific criteria. So next is the program selection. So 3 programs, the letter M and table 1, with different speed and sensitivity are available for nucleotide versus nucleotide sequences alignment. So the default, the MegaBLAST first is better for certain tasks such as identifying the input query and searching with large genomic query. The second is the discontiguous MegaBLAST. It works better than finding related sequences from other organisms, while BLASTn works better for short input queries and identifying short matches. So it also works better for cross-species searches than MegaBLAST. Next is the letter N. So by clicking the BLAST button, it submits the search to BLAST server for processing. Results will be automatically displayed when completed. And the last is the algorithm parameters. Algorithm parameters link opens a normally collapsed section that allows access to other parameter settings, including adjustment of number of alignments saved, search stringency, scoring systems or score matrix, and gap penalties, as well as query filtering. Detailed description are given next. Next is the general parameters. So parameters in this section specify the search sensitivity. So the first is the max target sequences. So it is sets the maximum database matches BLAST saves for a query. Second is the letter B, the check short queries checkbox. So it allows BLAST to automatically optimize settings for short input queries of 50 nucleotides or less. So next is the expect threshold. So filters out matches that are less significant with expect value above the settings. So the next is the letter D, the word size. So it is a set the size of the initial seed match phase. Smaller settings are more sensitive. And next is the letter A, the max matches in a query range. So it is the limits the matches saved to a given region of the query, such as form repeats. So matches to other region of the query are not crowded out. So the default setting of 0 means no limit. Next is the scoring parameters. So parameters in this section specify the search sensitivity. So first is the letter F, match or mismatch scores. So it is specifies the reward assigned to exact match and penalty assigned to a mismatch. So the next is the gap cost, the letter G. So field specifies how gaps introduced in the alignment should be penalized. So for Mega BLAST, the default is linear, no penalty for opening a gap, while extending a gap assumes a linear penalty proportional to the length of the gap. So for both parameters, our non-default settings can be selected using the pull-down menu. So next is the filters and masking. So first is the letter H. So parameters in the section specify whether low complexity sequences and organism specific repeats should be filtered. So next, the letter I. So mask for lookup table only. So whether to filter only at the initial match stage or during the alignment extension as well. So in lowercase letters in the query, so or provide us a mix upper and lowercase letters in FASTA. So it representing custom features and can also be masked. So that's all. Thank you.